Thank you everyone for taking the time to be here. Welcome, iMediSync friends and family. We're really excited to have Jay Gunkelman and Karosh speak to us today on neuromarkers and their implications for ADHD treatment. This presentation is co-sponsored by Rostrial and iMediSync. All right. Actually, we need to back up a few.
My apologies. Here we go.
Oh, we're good. All right. So a little bit about us. We are AI-driven mental health platform. We utilize dry EEG measurement technology. This is our beautiful device here. And we have not one, but 19 amplifiers. And we're able to integrate neuroimaging with HRV, heart rate variability measurements. And we offer preventive monitoring and evidence-based care. Okay. So our iMediSync team North America consists of Cody Newman, Chris Che, Carlos Chinchilla, and myself, Rebecca Walker. And we're a very sociable group, so please find us on LinkedIn or send us an email if you have any questions or ideas. We love to hear from you. We also have our CEO, Dr. Kong, who's the mastermind behind it. As I mentioned, we have Dr. Kim, who's our head of research and development. Every time I listen to him speak, I learn something new about what's been researched and studied. And ours truly, Dave Burkus, who helps oversee North America.
Para o pessoal do Brasil, a Rastreal é a distribuidora master da tecnologia em todo o território nacional brasileiro. A gente vem colaborando com a iMediSync na implementação dessa tecnologia aí no Brasil. Nosso objetivo é mostrar que a iMediSync, ela não vai, qualquer tipo de IA, ela não vai substituir qualquer tipo de trabalho médico ou de psicólogos, e sim colaborar com um diagnóstico mais assertivo e detalhado, tanto para entender o que acontece no cérebro dos pacientes, quanto também acompanhar aí o tratamento medicamentoso desses pacientes. É um prazer ter vocês aqui do Brasil conosco hoje.
All right, thank you, Hugo. So we have our device, which is the iSync Wave, which is what I showed you. And that uses our AI integrated mental care platform. We use a cloud, so it's wireless, and you can get your results back within 10 minutes of uploading the scan. And then we have iSync Brain, which is the software side. We have an age and sex matched database, as well as AI denoising and comprehensive quantitative reports. And I'll also mention that we're just coming out with a new report that is a mental health checkup that assesses things like ADHD, anxiety, depression, and says the probability of you having it and to seek further guidance. And so that's something new that's coming down the pipeline. All right. Our areas of use are for mental health, pediatrics, autism, as well as in research and medical offices. And we love connecting with all of you. So if you haven't subscribed to our newsletter, please do so on our website. You can also schedule a demo of how our device works and all the multiple reports that are generated from it. And there's also our LinkedIn QR code. And before I turn the time over to our presenters, we do have a presentation in November, mid-November, for ADHD and autism using red light therapy by Linda Thompson and her associate. So without further ado, I turn the floor over to Dr. Karosh.
Good afternoon, everyone. Thank you, Rebecca. Thank you, the team, for making this happen. And of course, thank you to Jay, my partner in crime. in helping put this presentation together. So today we're going to talk about ADHD and ADD. Now, before I progress with the slides, I want everyone to just think for a moment of a client or someone you have known, maybe a loved one, that has struggled with ADD or ADHD. And just for a moment, bring that kind of person to mind and see what kind of things you recall or you observed from that person in terms of their behavior, how they are thinking, anything that you could associate with the profile. All right. So now we're going to jump into the first slide. So ADHD or ADD, basically, it's one of the most difficult things to sometimes narrow down because there are a number of comorbidities that are oftentimes associated with these two conditions. On top of that, we have basically kind of an iceberg model where we don't really often see this symptoms. So obviously, sometimes these symptoms are hidden, sometimes these are not apparent, in the form of a kid running around being hyperactive or someone being in the daydreaming mode, having their heads in the clouds. So in a nutshell, when we define ADHD, we talk about difficulty with attention, hyperactivity, fidgeting, and impulsivity. But if we go below the surface of this, you can see that the number of issues is quite profound. You can see that it's associated with sleeping, for example, and Jay will talk about this in a moment, or the inability to regulate emotions, sometimes having a difficult time to bring down the intensity of the emotion. And we'll see that in a couple of slides down. But if you look at this massive list of all the possible things that can be a problem in ADHD, you recognize that individuals suffering from ADD or ADHD really have quite a bit to deal with. So attention is maybe one small aspect, but how does this really translate into everyday functioning? And we know in some of the adults that we've encountered who have ADHD or ADD, they have a very tough time going about their job or their social interactions. And so it is quite the condition. Now, let's go through the DSM-5, and I know we have ICD-11 as well for my international colleagues. So in North America, basically, per DSM-5, which is the latest version of our Bible for psychological and psychiatric disorders, the main things that they talk about are difficulty with attention, so sustaining attention, or basically even initiating attention, having a difficult time focusing at one task at a time. And you can see how often that could be a problem if somebody is at a job and they're trying to finish something on time. The other part is prioritizing tasks. So in part of it, of course, is the executive brain that has a difficult time with figuring out what is actually important and what is less important, and really grasping that and working towards that. Losing things, this is oftentimes seen in kids, but how often do you see someone who, as an adult, loses important stuff to them, iPhones and keys, etc. Distractibility, if somebody has ADHD or ADD, it's very difficult to shut out external stimuli and focus on what's going on in front of them. And the list goes on and on for basically what we call this inattentive subtype. And oftentimes there is this element of ADD that is persevering in adults. So as children or adolescents, we oftentimes see the hyperactive or the impulsive subtype. When they grow up to become adults, the one thing that really remains is that inattentiveness. And the hyperactivity is usually remaining if the person has any kind of vigilance regulation issues. They have basically impulsivity that's left of their childhood. So the next one, of course, is the hyperactive or the impulsive subtype. And, again, as the DSM criteria are describing nicely here, having a difficult time with vigilance regulation and having a tough time with impulse control, with remaining rested. They basically have an engine working inside them, and you've seen sometimes your colleagues who cannot sit still and they have to get moving. In children, it's more obvious because oftentimes they wear their impulsivity and hyperactivity. And if you treat children, you can see them calling the walls in your office. Oftentimes, one of the things that is easily missed is the blurting out of answers and the difficulty waiting the turn. And this is seen also in adults. And as I mentioned before, delayed sleep is a difficult feature, and Jay will talk about some of the sleep cycle issues that we see in ADHD. So we can have the hyperactive impulsive subtype, or we can have the inattentive subtype, or they could have a mixed presentation. So next slide, please. One of the models that we use basically to describe the two different subtypes of ADHD, so whether it's ADD, so the inattentive subtype, or the hyperactive and impulsive subtype, is called the vigilance model. And really what the vigilance model is showing us is the ability to regulate vigilance, and that's basically it. If you look at the hyperactive or the impulsive subtype, you can see that they have a difficulty with keeping themselves basically from regulating their sleep. So that's where actually the model comes from. Vigilance model comes from the ability to regulate falling asleep or actually staying awake. So either one of them is basically your ability to keep vigilance. Now, especially in children, one of the things that we see is they have difficulty with regulating the vigilance, and we call that auto-stabilization syndrome. So basically, these children keep moving so that they can keep themselves awake. And this you can see in their hyperactivity. They're oftentimes sensation seeking, or they're over talking. And you can see the same elements basically in adults minus the hyperactivity. So adults can also be sensation seeking or quite talkative, as we talked about in DSM-5, where the talking out of turn can happen. That's again the talkativeness. And then the other one, of course, is looking at cognitive deficits in the vigilance model. And this typically describes the inattentive subtypes because they have a difficult time sustaining their attention. And so when we look at the kind of the middle ground, we can get basically the combined subtype. Next slide, please. All right, so the history of ADHD, they used to call this the hyperkinetic reaction of childhood in DSM-2 back in the days in 1968. We now know that it's not just a condition for children, and actually many adults are having a tough go with ADHD. The latest one, of course, is in 2013. I'm not going to go through every single one of these historical iterations, but the latest one that we know, we have basically a description of 3 different types or subtypes. So the inattentive, the hyperactive and impulsive, and the combined. Now, there are also some individuals, researchers out there who have more subtypes that they describe through their work in imaging studies. And if you're interested, Dr. Daniel Amen is very nicely describing some of the things he has observed in his subtypes. They're not DSM-5, but nonetheless, he shows some evidence that even within these three subtypes, there are more subtypes present. Okay, so the next slide. Now we know that in the US, there are major social implications for ADHD, and that's worldwide as well. I live in Canada. We have very similar numbers. Just for adolescents, we know about 9.5% have been officially diagnosed. These are CDC numbers. There's also quite a bit of pushback, even though we've had about 40 years of data on the neurological aspects of this condition. And we see, unfortunately, there are many invested interests that don't want necessarily to recognize that we actually know what's going on neurologically. And Jay will talk about the neurobiology in a moment. Also, just to give you a teaser example, in the Boulder County, the number of ADHD cases basically is higher than all the minority groups, except the Hispanics. So it could be quite high in some counties in the U.S. We have basically many. I'm sorry?
Oh,
Okay, thank you. So we also know that a lot of issues with children who suffer from ADHD comes from being ostracized, and many times these children hear negative comments, and their behavior is ostracized. The way whether it's the educators in school, or whether it's other parents, or whether it's other kids. And so the impact on the self-esteem is quite profound. We know that another issue in teenage pregnancy is ADHD. So we know that this is associated with 62% of adolescent teen males becoming father by age 19, and 34% of females of this group by age 19. So one of the things that we know is families of children with ADHD have a lot of emotional stress, because you can imagine if the parents are being called every other day to school, and if there are difficulties between the parents figuring out how to deal with these children. On top of that, of course, genetics play a role. So sometimes we forget that parents of children with ADHD also may have ADHD themselves. So the likelihood of marital conflict is high, and we see that 3 times they're more likely to get divorced. And we know that adults with ADHD themselves, there is a 15 times divorce rate of the average population. And the reason for this, of course, going back, we know that impulse control is an issue. And oftentimes we see infidelity quite a bit happen in ADHD, in the adult population suffering from ADHD. The CDC basically estimates that in terms of healthcare costs, there is a $31.6 billion that we spend more on healthcare related costs and work absence due to ADHD. The prison population has a high prevalence of ADHD, about 21 to 45%. And we know that, again, that is another expense, about $27 billion in basically extra expense that we have in our society. So if we actually look at ADHD costs, the number is absolutely insane. So $58.6 billion in ADHD costs. And if you put Amazon, YouTube, and Target's revenues all together, they don't even match that. Any questions so far? I just want to make sure that everyone's following the slides. All right. So one of the things that we know with ADHD and are associated features, now DSM doesn't necessarily list these, but they're quite important. So emotional impulsivity, I alluded to that, is one of the issues. And basically, it's an aspect of poor inhibition, and this is typically associated with ADHD. To people who suffer from impulse control issues, have low frustration tolerance, they generally react quickly. You can see that children can get really angry, even adults, they lose their temper very quickly, aggression, and oftentimes they can have general emotional excitability that happens quite rapidly. And what we know is they don't have the capacity to delay a response to an external stimuli as easily as the general population. And also they have something called deficient emotional self-regulation, which basically means that the ability to self-soothe is not oftentimes there. And it's something else that we notice is somatic reactions that are oftentimes also there. So we see that they can get their blood pressure go up, their heart rate go up, and the body also very much responds as quickly to external stimuli. And a lot of this is basically going back to the structures in the brain that we know of. So the frontal lobe, the anterior cingulate, the ventral striatum, and the amygdala. These are the things that we know are involved in executive, basically circuitry. And we know that some of these, specifically the amygdala control and the limbic system controls, are not always working at the best that they can. All right, so I'm going to hand off to Jay now.
All right. Thank you, Karosh.
Thank you, Jay.
There's a percentage of the ADD population that actually have nocturnal sleep problems, circadian rhythm issues, and their inability to sleep well at night ends up making them drowsy in the classroom, and they end up being classified as inattentive. If you fix their sleep disorder, they can end up having normal attentional skill set, but they have to be able to sleep. So the sleep phenotypes are identified. One of the major endophenotypic clusters are people that have, it's like narcolepsy, but no cataplexy. They have a primary disorder of vigilance. They literally cannot stay awake. If you sit them in a desk in a school setting, they'll doze off in the desk. And for those individuals and only those individuals, they don't respond to the normal stimulants because they're not under aroused. They have a vigilance issue and they cannot maintain a vigilant state. And for those individuals, Provigil, which is modafinil, ends up being a medication that's used for narcolepsy. But for those that have a primary disorder of vigilance, it's like narcolepsy with no cataplexy. They have a forced sleep. As long as they're not moving or staying very active, they doze right off. There's a group of sleep disorders that are referred to as delayed circadian rhythm, and they can be treated with bright lights in the morning. Well, on the right here, you see the photoelectric industry in the United States has identified the desert Southwest as having intense sunlight. And the intense sunlight is bright light treatment in the morning, basically. So if you look at the co-registration with the map on the right with the map on the left, the very light colored states in the desert Southwest have about a 10% smaller incidence of ADD. The overall incidence is 9.5%, but if you look, some of the states are up into the teens, 14 to 17%, and some are down 5 to 7%. The desert Southwest essentially is treating delayed circadian rhythm, even though they don't necessarily realize that that's what they're doing, that they're just being exposed to the normal light. Minnesota and Illinois calculate their incidence of ADD differently than the rest. These are all classic CDC definitions, except Minnesota and Illinois. So we really have to get beyond a simple DSM description of what's going on. We really have to see into the brain in order to understand what's really going on in ADD. Simply looking at the behavior, which is the DSM approach, really misses the core of it. So let's look at something under the hood. We're going to be looking at EEG, which is electrical activity in the cortex that's being recorded in the EEG, and each of the channels in the waveforms end up coming from a different electrode, and those, when added up, end up giving us 19 channels, which are recorded from the iSync wave device. And they basically are going to give us a mapping. Now, the EEG is the raw waveforms, but the raw waveforms are specialized. People like myself look at the raw waveforms and see what's in them, but it's not easy. It takes some specialized training to read the raw waveforms. Luckily, we have the computerized quantitative analysis that takes the raw waveforms and breaks them down with independent component analysis into the pieces that make up the overall EEG. And when you break it down into the individual pieces, there's the raw waveforms, there's the spectra, which go from low frequencies to high frequencies, and we'll look at those frequencies in a moment, the topography of where the spectra is coming from, and then source analysis to point to where the source of the surface topography is coming from. And all of this is done within that 10 minutes that from the time you finish the recording to the time you get report back, this all comes back to you in a package report. EEG is broken into bands. The bands have frequencies associated with them, from very low frequencies, from essentially no oscillation below 1 hertz, up to 4 cycles a second, is called delta. Delta rhythms are seen in early life. A newborn has a lot of very slow content, but as you mature up into the 5, 6, 7 years old, the background isn't dominated by delta anymore. You end up generating faster frequencies. So you can see some of the correlates of the various EEG bands on the right. Theta is slightly faster, from 4 to 8, and definitions and frequencies sometimes vary between labs, but these are kind of classical definitions. And the 4 to 8 ends up being limbic system, but also occasionally a pathological slowing of the alpha, and that'll be identified again by looking at the EEG and the QEEG together. Alpha frequencies, there's alpha 1 and alpha 2. 8 to 10 is alpha 1, 10 to 12 is alpha 2. This faster alphas, better semantic and declarative memory performance, and if your alpha is slower, you don't have as good a memory performance. And they've actually shown that it's not just a correlation, that if you take somebody with slow alpha and you speed it up with a treatment, that you actually improve the semantic and declarative memory performance. So it's a causal relationship between the alpha frequency and memory performance. And obviously, attentional problems can be seen with alpha up front because alpha is a resting state and the frontal lobe should be busy. When you close your eyes, you see alpha in the back of the head, but the front of the head should actually still be busy. You just because you close your eyes doesn't mean the executive control frontally can take a break. The visual system at the back should be turned into an idling condition, but not necessarily the front. So when you see frontal alpha, it's problematic. Frontal theta is also problematic. We'll look at those mechanisms in a little bit. Sensory motor rhythm is a specific band range from 12 to 15 that was identified by Barry Sturman at UCLA, initially in cats, but they identified it in humans as well. And they find that you can stop seizures by training SMR, and you can also do things like brain brightening, speeding up the background alpha rhythm by training some of the SMR band activity, and getting again a brain brightening enhancement of neurocognitive function. They've shown that it helps with sleep and also memory consolidation during sleep. Beta frequencies go from lower beta frequencies to faster beta frequencies, but beta's the cortex busy doing its thing. The lower beta frequencies are quite often task related. The higher frequencies of beta are usually cortical hyperexcitability. Gamma's generated when you have neural networks that are formed. Gamma is a resonant property of having a network that's formed. And obviously, these networks forming as neural network dynamics is the ability of the brain to actually do the work that the brain has. So all of these frequencies are actually present at the same time in different amounts. And that's why we have a frequency spectra that shows the frequency and power within each of the frequency bands. So Karosha is going to discuss a clinical case, and then I'll show the EEG for that case.
On mute. Okay, there we go. So our first clinical case is RF, and I know it says at the title theta, but this gentleman also had difficulty with what we call spindling beta, and Jay will show you what that looks like. So this is a case that we saw at our center as a 42-year-old male engineer, difficulty with attention, basically whether it was initiating it or sustaining it. Oftentimes, and you can imagine how difficult this is for someone who's an engineer to do projects with the background of these difficulties. So he was procrastinating on projects, oftentimes not finishing the work in front of him. He had the ants in his pants kind of situation where he had to be constantly moving, couldn't stay behind his computer and finish the task. Talkative, despite being an introvert. Now, this is an important point because sometimes we assume that people only who are extroverted, are talkative. But someone who's an introverted person and has ADHD can also have that quality because they really cannot control that impulse that they have to talk. Easily distractible. And again, this is an engineer, so imagine trying to finish something. Time management was an issue. He had quite a low self-esteem, surprisingly as well, despite being a very bright and successful individual. And that's because his work was often picked apart by his boss for mistakes. And I cannot imagine what kind of mistakes, but in engineering, I assume you have to be pretty precise. So it really wears you down if you have somebody picking on you all the time. Struggle with anxiety. This is where the beta stuff also comes in. So overthinking every social situation, and also lying awake, worrying about everything and everything. And finally, as I mentioned, he had difficulty with confidence. Back to you, Jay.
So we're going to jump out of the PowerPoint, and I'm going to show you some EEG. And this is the RF case with theta and beta in it. I'm going to start out with the eyes open, raw EEG. And 1 second goes between the dark lines. 200 milliseconds is the lighter lines that you see. And obviously a wave that one moment. I should be able to share my screen. There, you see? So between the dark lines is 1 second. Thank you for the jog of the memory here. As we scroll through the EEG, you'll see some waves that take 1/5 of a second to happen. That's 1, 2, 3, 4, 5 to 6 of them happening in a second. That puts it in the theta frequency range, if you remember the 4 to 8. So 5 or 6 puts it into the theta band. You can see, obviously, this is a sinusoidal rhythm. 1, 2, 3, 4, 5, approximately 6 of them happening. But if you look very carefully, there's little tiny fast waves going on as well. And as we scroll through the EEG, we're going to see more of the spindling fast activity. Pardon the dog in the background. The spindling beta that you see in here is hyperexcitable cortex, and the theta up front is less perfusion up front, less activity in the front. So we've got fast and slow happening at the same time. There's actually an ideal paper to describe the treatment of this case. Sandra Loo, L-O-O, Sandra Loo at UCLA's Semel Research published a nice study on ADD. And it basically says if you have frontal-central theta, and you can see frontal and central, the theta activity. Frontal electrodes, central electrodes. So frontal-central theta suggests a dopamine deficit in the striatum, and it's treated with methylphenidate, which is a dopamine reuptake inhibitor. But the beta is treated with the guanfacine. Guanfacine reduces norepinephrine peripherally and centrally. And peripherally, obviously, it's a blood pressure medication, but centrally, it reduces beta in the EEG. And in her paper, she says if you have both fast and slow, you give both medications. So it's not an easy guess to say, well, I need to get rid of beta and theta, because if you don't look at the EEG, you don't know it's there. Behaviorally, this looks just like regular ADD, but there's specific meds that match the specific EEG patterns. Now, I'm going to jump out of this, and this is the eyes open. Oops, wrong one. This is the eyes open normative, and this is from the iSync database. These are z-scores. Normal is green in the middle. Too much is red. Too little is blue. Now I've filtered out the low frequencies below 1.6, so there's no 1 hertz in it. That's due to the filter I used, but you can see the excess in the theta band: 4, 5, 6, 7, 8 hertz. C3, C4 in the, we didn't mention the phenomenon of mu, but mu is C3, C4 alpha frequencies, and there's some of that in this. And then there's beta, and you can see the beta as an excess going up into higher frequencies. This is absolute power. Actually, looking at the microvolt square and the z-scores of that. We can also look at relative power, which takes into account skull thickness, and you can see the theta is in excess, the mu centrally, which tells us the frontal lobe is not engaged, the mirror neuron's not engaged, and we've got theta up front to account for that, the reason for the mu. And then you can see the beta spindles in this. And without the excess of the absolute power, we can actually see the sources a little bit more clearly, in this case with relative power. Both of them are available in the database. To look a little bit more in detail at what the database has in it, it's got a little index at the start, but this is the raw EEG, and I have to bump up the size of this, but this is the EEG and this is the EEG cleaned. And you can scroll through the EEG. It's broken into components, which are the pieces that make it up. And one of the biggest components is full of 4 to 8 theta and beta. And the source of that mixture of frequencies ends up being seen as something more upfront. You can see the source analysis puts the location a bit more right frontally than left frontally, which we see in the topography. So there's 19 of these, and each of those are pieces that make up the overall EEG. And in component analysis, you can take components out. This is a mu component on the right side, for instance. You can see it's in the alpha band and it's right central. There'll be another one similar to it on the left side. It's again mu on the left side. Now, again, 19 pieces that make up the EEG. Then the delta, theta, alpha, and beta broad bands, and these are again z-scores. Then and relative there. Then 1 hertz at a time, so you can see in high resolution. Now, there's a lot more in the database, but for us to describe ADD, ADHD, this theta and beta in this case end up describing this particular case. You can see each of the electrodes I call this a Hollywood square display. Each electrode has its own spot here. FP1, FP2, left frontal, right frontal, above the left ear, above the right ear, at the back of the head. So this is laid out like you're looking down, ears on the side and the nose up front. And red is too much and blue is not enough. And some of the very, very high frequencies are sort of missing, but the excess of the theta and the excess of the beta, and where they're seen in excess. So the topography is there, and it's seen in absolute 10. This is in decibels per hertz. This is a log transform that corrects for 1 over f, so you can end up seeing high frequencies and low frequencies more easily. And here you see the theta and the beta. And this is a display showing the statistics for the alpha band. 8 to 12 alpha frequencies are important in a lot of different disorders. And in ADD, the tuning of alpha may be critical for identifying somebody that needs an amphetamine. We'll look at that later. If you have very slow alpha, the amphetamines give you a little bit of norepinephrine and speed up the background alpha. So this says the peak power is 2.3 microvolts, and the z-score is basically at the pretty close to the middle of the z-score band. My dog's going to probably get up and sit on my lap here. We'll see. The z-scores basically say this is a normal amount, and it's tuned in frequency. It's 9.8 hertz at the peak, and that's pretty much just a slight bit slower than the norm, but it's well within the 1 z-score. So it's normal alpha left, as normal alpha on the right. It's alpha greater left than right is a asymmetry, which we'll look at as a separate feature. But there's a lot of additional detail in the database, connectivity issues, source level connectivity. We're not going to get into this at this time, but there's source connectivity for research that you're going to be doing perhaps on various things. There's default mode network connectivity specifically as well. So there's a lot of pieces that are seen in the database, and not all of them are going to be used for the ADD, ADHD display basically that we're doing now. That was an eyes closed up above, and we're going through the same basic thing below for the eyes closed. So the norms are really quite striking. This is the eyes closed ADD. This is the eyes closed ADD for this case. And it looks very much like the eyes open. This is actually a decreased vigilance moment. We talked about vigilance, and when you start out with alpha and then it slows and theta becomes more prominent, this is less vigilant. Alpha comes back, they're more vigilant. And if you stay mobile, you maintain all the faster content. But when you sit still, it took this person less than 30 seconds to get into a drowsy state. So the eyes closed is important. If we see a vertex sharp wave of sleep before 300 seconds, it means they have to go to a sleep lab. And the mixture of fast and slow ends up being identifiable in this case. You can see the theta, you can see the beta. The beta works as a vigilance mechanism. At CZ, it wakes you up. And for people that have insomnia, usually CZ beta is part of the problem. So,
Jay, I have a question from Simona. What if the alpha peak 0102 is 9.2 hertz?
Well, it depends upon age, obviously, and age and sex end up matching up with the database, and they'll tell you exactly how many z-scores off you might be. So it's quantitatively answered, but usually 9.2 is going to be in the alpha band nicely. It's not at the bottom edge near 8. It's not at the upper edge near 12. It's kind of Goldilocks frequency between 9 and 10. So that's a perfectly appropriate frequency. What I'm going to do is to stop the screen sharing of this EEG, and I'm going to go back to the PowerPoint as a screen share. And oops, stop, and I have to pick a different underlying screen here. And so we're back at the PowerPoint, and I need to get this back to projection. And so Karosha is back on, describing his second case, case EP.
All right, so this one we saw it spindling beta already, and we saw the impact of it in a previous case. Now this one is a 50-year-old female social worker. So she misses out on details. And again, I just want you to think of functionally how this would look like if you're working as a social worker and you have to listen to your clients and not always picking up all the details. So hard time staying on task, again, mismanagement of her time, forgetting appointments. So that's a big one. Can't really get a grip of a task due to poor organization. She and does not like anything that it requires listening to instructions. She's always hearing her husband complain about losing keys and important stuff, interrupting others mid-conversation. So as a social worker, you need to be able to listen to people, and not always that easy for her when she's interrupting them. Again, difficulty with emotion regulation, so gets easily annoyed, reactivity to external stimuli is high for her. She spends the money that she doesn't have, and also she basically wants to be the life of the party at times. The impulse control issues, and this is something that we talked about in terms of percent of marital basically difficulties, which was above 15 times the norm, and she had an affair in her relationship. So Jay is going to present the case. I think Jay is attending to her puppy, to his puppy, somewhere there.
I am back. A Rhodesian Ridgeback, he needs some attention. So let me stop the screen share and pop up the EEG. So back to, this is the, well, it's going to be the eyes open EEG here. And share the screen. So right from the very beginning of the eyes open EEG, we don't really see alpha at the back of the head, but we see spindling beta. 1, 2, 3, 4, 5.5. Well, 5 times 5 is 25. This is in the upper 20s somewhere. And as if you're used to looking at a lot of EEGs, there's a lot of beta in this. And it's spindling excess beta. It's not all muscle. A lot of people will mistake spindling beta from the EEG as a muscle artifact, but it's sinusoidal. Muscle is a biphasic spike, a point on the top and the bottom of the waveform, and these are sinusoidal. So this has excess beta in it, and I'm going to pop in the eyes closed. This is the same case with the eyes closed. We expect with the eyes closed to see alpha at the back of the head. There's a little burst of alpha, but it doesn't sustain itself. We expect alpha to be, with the eyes closed, a persistent presence at the back. You see instead beta spindling at CZ, C3, C4, and beta up front. CZ beta spindles a classic for insomnia, and it's also present in disturbed sleep. So individuals that have difficulty maintaining vigilance, this is a burst of alpha. And usually as people start to get drowsy that have the beta spindle excess, they'll have a burst of alpha and they'll slip into a drowsier state. You can see how slow this is. 1, 2, this is slipping into the delta band, but it's rhythmic slow activity at CZ. And this person is going to wake back up with spindling beta. You see, the drowsiness brought them back to an awake state with the beta spindles. So if you have beta spindling as an issue, you need to end up having the beta spindles dealt with. Not everybody that has ADD ends up having beta spindles. It's actually one of the clusters. We'll look at the various subclusters and subtypes. Let me jump to this and show the eyes open. This jumps straight to the spectra. Again, z-scores, a deficit is blue and excess is red. You can see the excess in the beta band. This is 19, 20, 21, 22. So an excess of spindling beta centrally. CZ is the max, but we counted on the raw waveforms, some of them up into the upper 20s, and you can see it goes all the way up into the 20s, from the teens to the 20s. Absolute, relative, because relative is always relative. And if you're the only one who's got any, you become the giant when you go to relative. There's a little bit of slower stuff in the EEG, but this is dominantly spindling excess beta. The alpha band stops at 12, and the SMR frequency is 12 to 15. But above that, you end up starting to see the excesses statistically mount up. I'm going to jump back. This is the eyes closed. We won't scroll through the entire report to get there, but you can see in absolute, C3, C4, and CZ light up. And relative, again, relative shows the beta spindling. 1 over F, the faster you are, the smaller you are. It's a basic rule with EEG. And beta is inherently small because it's fast. And in absolute power, quite often the lower frequencies will dominate, and you may not see the beta until you get to relative. But in this case, because it's the dominant entity, it dominates the relative power. I'm not going to go through any of the other EEG stuff because we've got a lot of other material to cover. And let me get back to the PowerPoint here. So project this. So we've got spindling excess beta and those. There's one other raw EEG that we're going to take a quick look at, and this one has a spike in it. Specifically, there was a recent meta-analysis that looked at spike activity in across a wide variety of different conditions, and in the autism spectrum, for instance, between 50 and 70% of the autism spectrum ended up having epileptogenic-looking potentials. Spike activity in the EEG is usually considered epileptogenic. It's something that could cause epilepsy. It doesn't always cause epilepsy. Sometimes it simply creates a discharge that causes a tic. And one of the common comorbidities with ADD is Tourette's. And the clinical case that I'm going to show, and let me again jump out of this. My apologies for jumping back and forth. I have to jump out of that to get to this. And so this is the Tourette case. And I'm just about to share that. So there. So this is the start of the EEG at 1, 2, 3, 4 seconds in. And as we scroll through the EEG, there is a spike that shows up. This is a spike. Now, this one's positive, electropositive is down in EEG, and this spike and slow wave are coming out of the temporal area. This is a person who had vocalizations and tics. And we'll look at the source of tics as being the caudate, and we'll look at that structure in a little bit. But the spike coming out of the temporal lobe, spikes don't happen all the time. They're transient. This one happened only a few seconds into the EEG, but it repeats itself. Spikes don't always show themselves a lot. but you can see another spike here. And as we continue through, a little bit of artifact from muscle in the temporal area. And here's a spike, a little slow feature that got cut out, but you can see the spike again and again and again. So this patient actually had tics and vocalizations, and the caudate, the head of the caudate's involved in motor tics, but the tail of the caudate hooks into the stria terminalis and the temporal lobe limbic structures. And we see this in the temporal area, not up front associated with the motor tic, but in the temporal area generally associated with the, again, limbic speech, which you don't need to demonstrate, guttural utterances, basically, and also animal sounds and throat clearing and those kinds of sounds that are tick-like. Anyway, this is seen in the EEG. Spikes are not usually the thing that you look at with normal quantitative methods. They are best seen in the raw EEG, which is one of the reasons that they have people like me still around looking at raw waveforms. So this is a Tourette case that has comorbid spike activity. When you see a spike, they average out when you average things together. They need to be looked at more specifically. There's software that has spike detection and things like that in it, but you need to be aware. In the ADD population, historically, people were saying that the incidence was closer to 10%, but the meta-analysis that was just done looked at large studies, and they found about a third of the ADD population ended up having small spikes in their EEG. And when you have spikes in your EEG, usually an anticonvulsant is going to be involved in the treatment. So that's it for the raw EEG aspects of the sharing here. And we should be back at the, so do you see these, the phenotype page? Basically, there's 11 phenotypes, and a phenotype is an endophenotype. It's between genetics and behavior, there's an intermediate step, and it's the phenotypes or endophenotypes. And so they're genetically linked, but genes sometimes don't express themselves, but phenotypes are expressed genetics, and the phenotypic patterns end up predicting medication and other therapeutic interventions. So what we basically end up looking at is the psychophysiological expression of the genetics that's actually a mixture of nature, the genetics, and nurture. How is it going to express itself? So these are the 11 endophenotypic patterns. Low voltage fast EEG actually has it's the EEG without any rhythm in it. Adrian and Matthews were one of the first groups after the EEG was originally discovered to actually look at it. And Adrian had alpha and Matthews had an EEG with no apparent rhythmicity, a low voltage fast EEG, which is declared a normal variant. Obviously, if you're lab partners, you don't call one of them abnormal. But it's a rare pattern. About 10% of the population have a low voltage fast EEG. It's associated with gene number 4. It's a well-known genetic correlate, and it's an endophenotypic pattern. Gene number 4 controls GABA receptors. When you produce more GABA receptors, then you can saturate. You're over-aroused, and you need more GABA. Well, you can either take something that gives you GABA, or alternatively, make it yourself. Alpha theta style training works very well with the low voltage fast EEG, and stimming at 10 hertz will give you some alpha with photobiomodulation with a low voltage. But we're here to talk about ADD and epileptiform content. We just looked at in an ADD as a comorbid feature. Again, about a third of them have it. Slow content in the EEG is usually white matter related, and you can have excess slow content in ADD. Very common to see frontal lobe hypoperfusion, either theta or alpha, which are both hypoperfusion. We'll look at the perfusion correlates in a moment. And then obviously spindling beta, which we've seen in the last two cases. So
can I just make a comment here on the epileptiform activity? So, Jay mentioned there's about 1/3 of these cases where they have epileptiform activity. And one of the biggest challenges, of course, is that these epileptiform activities lead to behavioral outbursts. So you actually um, see these kids who have, teachers complaining about their behavior in class. And oftentimes, of course, this is unknown to the teacher or the parents. Oftentimes, before they have these behavioral outbursts, they have an epileptiform activity. And the challenge, of course, is to identify cases like that and have them on a proper dose of anticonvulsant, which would likely work on reducing these behavioral outbursts.
temporal lobe discharges quite commonly cause not a convulsion because it's not motor, it's temporal. And temporal lobe epileptiform discharges have associated with what's called pseudobulbar affect, an emotion that does not make sense. And you feel free to ask questions. Just pop up.
This question's from Carolina, and it deals with epilepsy in the temporal lobe for someone who has epilepsy and ADHD. She asked, which is the more recommended PBM therapy specifically for that site?
In fact, we'll look at that. One of the things that photobiomodulation can do is cause a frequency following. So if you flash at a specific frequency, you tend to have more of that in the EEG. And we know that the SMR band, which we looked at very briefly in the list of frequencies, SMR is used to help with seizures. And the photobiomodulation in that frequency range gives you an SMR-related activity in the brain and ends up helping. So we'll look at that. And thanks for the question. What we see here is in the history of diagnosing ADD, theta beta ratio was looked at back in 1999. The theta beta ratio is Lubar, Monastra, and Linden. And that paper, if you were 3.5 as the ratio, 3.5 to 1, theta to beta, you were outside normal range at age 7. Now, the ratios norms drop down and down and down. By the time you're in your 20s, 1 to 1 is normal, and if you're much above 1.5 or 2, you're outside normal limits. So the age ends up influencing this. Kropotov looked in 2009 and found that to be abnormal required a 7 to 1. What's happened? Well, 1999 till now, kids get 2 hours less on the average sleep per night, and drowsiness facilitates theta. So the theta beta ratio is a higher ratio now than it used to be because the average kid now doesn't sleep as much as they historically had. Again, 2 hours a night is a lot of sleep to be missing. Frontal alpha is another thing that we can identify, and obviously frontal alpha or frontal theta ends up giving us a hypoperfusion frontally, and when the frontal lobe is not working as much or as well, those resting state EEG patterns end up showing up. People that have a sleep phenotypes, I mean, we mentioned these earlier. If you're unable to sleep properly at night, you end up with vigilance problems. And Martijn Arns and I published a paper on beta spindling and insomnia as a side effect of that. But he also followed up. We did it with 380 something people with a broad psychiatric swath. 49 of those were ADD. and the beta spindle was the predictor of insomnia. He did a second study with a more narrow population. About half of the study was ADD, and they found more impulsivity, hyperactivity, which were secondary findings in ours original study, but he found those to be more dominant. But then the population he was looking at was primarily ADD, again about half of them. So the ability to look at vigilance problems due to sleep issues is a big issue, and the beta spindling ends up being seen as a compensatory mechanism or a counter-regulatory mechanism. Reactive attachment, autism. There's all sorts of comorbid findings here. We basically end up having and then obviously subclinical seizures. And although it's listed here as 10%, again, the current study from 2022 with Martijn Arns et al. And myself included end up seeing about a 30% incidence in the ADD for epileptiform content. So this is a slow alpha variety. Alpha as a frequency in the back of the head, normally 8 to 12, can slow. And when it's slowed alpha, when you open and close the eyes, from eyes closed to eyes open, the alpha drops by about half. And when you see that change at a frequency that's in the theta band, it's slowed alpha. And slowed alpha can end up being very problematic because you need a fairly strong stimulant to speed you up out of the theta band back into the alpha band. And amphetamine will take slow alpha and make it back into the alpha that should be. It has a strong norepinephrine agonist effect, and that speeds up the background activity at a thalamic brainstem level, and that will fix the slow alpha. So this is eyes open. And when you've got widespread slow alpha eyes open, you're also less responsive to the outside world. You're going to look pretty inattentive and kind of drowsy. So gross under-arousal. How do you maintain activation if you've got something like this? Well, you're either moving, behavioral autoregulation, or you're taking an amphetamine as the proper medication. If this was endogenous theta, not slowed alpha, it would be methylphenidate. And we'll look at the medication selection issues in a little bit. This is simply excess theta up front. You've got C3, C4 mu in the image here. And this is frontal theta, again, in a z-score. This is an outside normal limits, excess theta up front. And again, C3, C4 is mu. Theta up front. If you have this live, you can spin these heads around and look at them in 3D. You can turn on one hemisphere only and look at the midline. With the software live, you can play with these images. This is normal on the left. This is the abnormal client on the right. So spindling excess beta. This is a pattern. This is in the eyes closed condition. This is delta, theta, alpha 1 and 2, beta 1, beta 2, beta 3, again, different frequencies, and then fast activity up in the gamma range. But this, an excess, statistical excess of beta. And that's one of the endophenotypic patterns. Spindling beta, you can see where the location is. So this is a paper that I did, one of the odd papers that I've done with no co-authors. And this basically looked at the EEG and the ability to predict medication using the EEG. Now, we didn't look at a specific clinical group because the phenotypes cut across the DSM categories, but we're looking at the phenotypes and biomarkers as predictors of medication. One of the things, this is work by Ian Cook out of UCLA's SEML Research, UCLA's NPI lab, and he's in private practice now, but he was the head of his department back then. This is perfusion, hyperperfusion, hypoperfusion. The mean statistical significance, 2 and 3 standard deviations of hypo and hyperperfusion. The theta and alpha band from about 4 up to about 11 or 12 ends up being hypoperfusion, but at about 11 up to about 16, you have a neutral positive perfusion. Remember, the SMR band is about 12 to 15. And at about 16 hertz, it takes off into hyperperfusion. Now, there's 3 lines. This is linked ears as a montage. Linked ears is not a good way to see local function. These are Laplacian techniques, which allow you to see local function very readily. So the hypoperfusion of the frontal lobe would be either theta or alpha, and then obviously hyperperfusion ends up being beta spindles. This is actually a Mac monkey brain. If you're wondering where the frontal lobe went, the stippled areas are suppressor strips. If you stimulate a suppressor area, you get less activity in the associated area. And one of them that I was particularly interested in looking at was 4S, the supplementary supramotor area, and for area 4, which is the motor strip. And the sensory motor strip ends up with the legs on the top, hands about there, and the face down at the bottom as you go down the homunculus. Well, the frontal lobe regulates the motor strip, and Niedermeyer wrote this nice paper about frontal lobe disconnection in ADD, and he didn't talk about what the circuitry was that disconnected. So when I found this in trying to find what the circuit would be, I was thinking, well, it's an easy circuit for S to 4. It's right there, just a cortical connection of some sort. But that's not reality. This is what happens. It goes from the front down to the caudate, to the putamen globus pallidus, to the thalamus, and then up to the motor strip. This network has a net excess of 1 GABA, so it's an inhibitory network. Now, what happens to this inhibitory network if you add the cingulate into this network? Well, it tosses, instead of going directly from the putamen globus pallidus thalamus, it goes putamen globus subthalamus thalamus, adding an extra negative GABA into this, making it a double negative. So the frontal lobe command stop, which would normally create a stoppage, becomes a stopping because the cingulate's involved in it. The perseveration associated with the cingulate is seen by basically adding an extra loop into this circuit, making it a perseverative loop. But the locations are important. The caudate is what causes the motor tics, and the tail of the caudate hooks into the temporal lobe. This is looking at the head of the caudate, where it's a motor function. The striatum operates with dopamine. The primary neurotransmitter here is dopamine. So if you have a DAT genetics, which is really a high incidence in the ADD population, it's dopamine transferase. It's an enzyme that strips away dopamine from the striatum. And if you end up having a problem here, you don't fix it with amphetamine to speed up the alpha. You deal with the dopamine deficit quite directly. We'll look at medication linkages in just a little bit. So that looked like it jumped all over the place. There's the caudate, putamen globus pallidus thalamus. They actually are fairly contiguous. This is a human dissection. Now, this is over 400 kids. Gordon Serfontaine in Australia and Bob Chabot at NYU were the collaborative authors on the original paper. But the original paper looked, and they found theta and alpha as 2 groups. Well, they then sorted out those 400 into groups that looked the same, and of the ones that had theta excess, some had beta excess as well. of the alpha excess. Some had beta as well. There's a group that was beta excess all by itself. Some looked normal at rest, but if you put them on a task, usually they show up with theta. And so you've got theta, alpha, and beta. How can that make sense? If you got theta or alpha or beta in the front, it gives you the same behavior. Well, when the front's not doing its job of attentional and affective regulation, you end up with a behavioral representation because the front's not doing its normal work. So whether it's a theta head or an alpha head or a beta head doesn't switch it from inattentive to, it's not a different type of ADD presentation clinically. The DSM does not identify the difference between these groups. So only when you look with the EEG do you identify, oh, it's excess theta or excess alpha, or perhaps the beta spindle pure type. It comes down to beta as a pure type being close to 10% when you look at it in other studies, and the mixed beta with other is another 10%. So beta compromises about 20% of the overall population, but again, as a pure type, a little under 10%. So these are, again, clusters where the EEG looks similar, and each of these with a theta in it are going to respond to a similar medication class, the ones with alpha to a similar medication class, and the beta spindle is pure type with a similar medication. So some of the medication prediction would come just directly from which one of these clusters it was in, but we can look in a more fine-grained manner. Here's the head of the caudate, the tail of the caudate here merging with the striatum terminalis, hooking into the amygdala and hippocampus. And again, motor tics are the head of the caudate, guttural utterances of the limbic system's amygdala, limbic speech or emotional speech, and then parroting echolalia is a hippocampal phenomenon, a memory phenomenon. So the tail of the caudate gives you the vocalizations, parroting, echolalia, or limbic speech. Words my grandmother would have me wash my mouth out with soap if I used. So what we've got is a complex brain. The frontal motor regulatory circuit ends up having a comorbidity with Tourette's because of the involvement of the caudate. This is actually an induced beta that comes on when you do a task. This is called a JTFA, joint time and frequency. And the JTFAs basically tell you at what point in time did what frequency light up with activity. This uses wavelets to tell you what's synchronous and what's not synchronous. Synchronous is hotter colors. Lack of synchrony is the colder colors. This is a normal person. This is a high-functioning ADD and a low-functioning ADD as measured with a CPT task. They're on the CPT task. They get the GO stimulus, and this is 1,000 milliseconds. So from the very beginning, when they get the stimulus, this is the spot that's going to light up in the brain. It's the spot that controls the right index finger because they're about to push a mouse button, and beta kicks in. This is the beta that kicks in. So about 500 milliseconds after the presentation of the GO stimulus, the person's brain recognizes the difference, and the person has reaction time from that point to this point that they hit the button. So it's about 500 milliseconds to there, another 120 milliseconds or so, and you get the button push. Now the low function, and by the way, you can see gamma, 1, 2, 3, 4, 5, number 6 bursts of gamma, 6 bursts of gamma in a second. That's gamma nested in theta. For those of you who are interested in cross-frequency coupling, this shows that the normal person has a lot more gamma than the ADD does. They have 1, 2, 3, 4, 5, 6, but they're very weak until the very end. The low-functioning ADD is missing some of the nested gamma. Gamma comes in chirps, brief bursts of gamma, not continuous gamma. Continuous gamma is pathology. Gamma that's healthy comes in brief chirps. And nested in theta. Chirps and nests, it sounds like a bunch of bird brains, but these are the terms that the field has come up with. So gamma is reduced, beta is more and more latent, and the time that it takes to respond is more and more latent as well. The low functioning ADD barely gets the button pushed by the end of the 1 second of time. So it's not like they're using a different circuit, but the ability to fire up the circuit and use it ends up being compromised. And there's the beta that happens on a task is not as intense. So stimulating in the beta range or training beta ends up being a way to work with the ADD population. And suppressing the theta and training up the beta was done originally in the early Monasterlin and Lubar papers. Since then, obviously alpha and beta deviations have been identified as well. This is a simple study that showed a baseline. This is neurofeedback where they train beta, resting state, train beta up, resting state, train beta up, resting state. The reason I put this up is that people will say they trained beta with neurofeedback and it didn't change. Others will say they trained beta and it got bigger. They're both correct. This is magnitude, which is the average, the integral average of the amplitude. The size of every single beta spindle is going to be small. Beta is inherently small, but you can make more packets of beta per unit time, so the magnitude goes up. So it's like midgets. Midgets aren't going to be big, but you can fill a room with midgets and you got more midgets. So what we have here is a room full of midgets and resting state fewer. So lots of little tiny beta packets and then fewer beta packets, more packets, fewer packets, more, fewer. So this is showing the operantly trained control of beta volitionally. Here's a spindle of beta. Normally, beta is a mixture of some very small fast activity, but spindling beta creates a sine wave. You can see it's frontal, all the F electrodes, FP1, FP2, frontal, F3, F4, kind of median frontal, F7, F8, lateral frontal, and FZ, dead center on the midline up front. Beta spindles frontally. And again, frontal beta spindles as a subset in the ADD population. And alpha frequencies and memory, we mentioned slow alpha and fast alpha being differential for memory function. This comes out of Wolfgang Klemisch's lab in Salzburg, Austria. There's a whole bunch of different classes of medication that can be given. And these are small print, but it colorful. You have to make meds identifiable one from the other, otherwise the pharmacist will get you really goofed up by giving you the right color of meds but the wrong drug. So that strength and color and everything is going to be differential. But let's get past this chart and actually look at things a little bit more. So we basically have a whole list of medications that you can see given at different points in time to different people with ADD effectively. Ritalin was one of the first stimulants that was used, and it's methylphenidate, and it's basically a dopamine reuptake inhibitor. So it's great for those that have a dopamine problem. Vyvanse or Adderall are an amphetamine, and they push norepinephrine and they speed up alpha. Strattera is a norepinephrine reuptake inhibitor, not an agonist like the amphetamines, but a reuptake inhibitor. They'll say it's not a stimulant. Well, it says stimulating properties of keeping the norepinephrine you make around with a reuptake. Provigil, we mentioned very briefly, this is an orexin agonist. Orexin is the wakefulness drive neurotransmitter. And orexin is going to increase beta spindling at CZ. People that have difficulty with not being able to stay awake needs more wakefulness drive. So the Provigil can be given to primary disorder of vigilance, which is again like a narcolepsy, forced sleep, inability to stay awake. If you're driving the car, you basically are going to end up having to pull to the side and fall asleep. And if you're walking on the sidewalk, you go over to the grass and lay down. I mean, it's forced sleep. So Provigil, if you're a student, you sit in your chair in the class and you fall asleep. And Provigil will keep you awake. Guanfacine and clonidine are channel blockers, alpha-2 agonists, and they reduce beta in the EEG. Guanfacine has been popularized for ADD. Clonidine has been popularized by Bessel van der Kolk for the PTSD population. Gabapentin also reduces beta, but it's an anticonvulsant. Now, it's given for anxiety, it's given for peripheral nerve pain. I mean, it's given for a lot of things. But one of the things that can be given for is beta spindling in the EEG that's more intense than that could be controlled with a simple channel blocker. Lamictal is also an anticonvulsant, and as is Depakote, they reduce beta, but they're also great if there's a paroxysm. Gabapentin is okay for beta spindles, but if it's a paroxysm that you see, it's usually more of a primary anticonvulsant. Gabapentin is an ancillary anticonvulsant, not a primary. It's given to cut down on the excitability of the cortex if the primary anticonvulsant needs a little help. Lithium. Well, lithium does cut down on beta, and it's an anti-manic compound, but it also potentiates discharges in the EEG. So if you have epileptiform content, lithium's contraindicated, as are the antipsychotics. Oxytocin. Well, what's oxytocin doing on a list for ADD? Well, if you have mu, mu's a mirror neuron disconnect. You're not relating to the outside world, and oxytocin is a social bonding hormone. So oxytocin reduces mu, and it increases, beta and alpha. So what we've got is a neurotransmitter that's not very commonly thought of as one that works. But if you've got this, disconnection of your mirror neuron system, and you've got rip-roaring big mu, oxytocin reduces that mu. Pardon me, I have diabetes insipidus. I have to keep drinking.
So Jay, I just wanted to make a comment here. I think on this list, you see a lot of medications that are basically off-label use. So in clinical practice in psychiatry, we don't oftentimes see all of these used. Some of them are, for example, the alpha-2 agonist, the stimulants, Cetera, which is a non-stimulant. But a lot of these medications that Jay is presenting, we can use them in ADHD off-label, and they are very effective. But this is something that you have to keep in mind if you're prescribing medications. You have to make sure that they are okay to be used off-label in your clinical population.
Obviously, an SSRI, SNRI, the serotonin reuptake inhibitor, and a serotonin and norepinephrine reuptake inhibitor. If it's regular alpha up front that's occupying the frontal lobe, an antidepressant like an SSRI will be very good. But if it's slow alpha up front, an SNRI to speed up the alpha and reduce it up front ends up being effective. And a tetracyclic is if your alpha is really fast. If you've got 10.5, 11, 12 hertz alpha up front, SSRIs are going to give an overarousal. SNRIs is going to be a big mistake, but a tetracyclic is like Remeron or trazodone end up being given at night they cut down on beta in the EEG, and they help you fall asleep. And they have an anti-anxiety effect during the day. And then obviously antipsychotics, this is Risperdal is more common in the ADD population than others, but many of them end up being prescribed. Now, as Karosh pointed out, some of these are on label. The rest of them are based on the EEG. If you have a phenotype match, you try to match the med with the phenotype. So how do you make the match? Methylphenidate's a dopamine reuptake inhibitor. If the theta in the EEG is seen, frontocentral theta, you've got dopamine deficit. So methylphenidate's a perfect match for frontocentral theta. And basically, if you've got frontocentral theta, methylphenidate's your match. Again, dopamine reuptake and a dopamine deficit. It's a perfect match, a lock and key. Amphetamines basically are norepinephrine agonists, and they speed up alpha. So if you have very slow alpha, an amphetamine will speed it up, and the person will function much better when alpha's in the alpha band instead of being in the theta band. Provigil, we've mentioned, it's an orexin agonist, and for somebody who cannot stay awake, a primary disorder of vigilance, is a nice match. Again, it's a stimulant form, but it's not like the other stimulants. And then Strattera is a norepinephrine reuptake inhibitor. And for slightly slow, you don't need an agonist, you just need a reuptake inhibitor. So for a slightly slow background alpha, Strattera is a good match. Now, clonidine and guanfacine channel blockers, again, reduce beta in the EEG. These are all beta spindle treatments, and clonidine and guanfacine reduce beta if it's a reasonable amount. If it's really intense beta, much bigger than just some excess, you may have to step up to gabapentin or pregabalin, which is Neurontin and Lyrica as brand names. But they reduce excitatory neurochemistry more efficiently or effectively than the clonidine or guanfacine. And if clonidine or guanfacine kind of work, but you think, well, I just need a little bit more power, you can't step up the dose on them easily because they do have a peripheral norepinephrine. They drop blood pressure, and you don't want a kid to stand up and pass out and fall over from low blood pressure. So if they're kind of working but not quite fully, you need to switch to a more potent class. And the ancillary anticonvulsants end up being that. Lithium, this is a very potent treatment for beta spindling, but it does potentiate epileptiform content. If there's a spike in the EEG, it'll make it worse. If there's a paroxysm in the EEG, it might make that into a spike. So if the EEG is not stable, then lithium is not your fix for beta treatment. Anticonvulsants, antipsychotics also potentiate discharges much more. And you'd think, well, there's some brand new antipsychotics. Aren't there some that work like they should without causing a problem? Actually, some of the newest are some of the worst. An antipsychotic or lithium can be given to reduce the beta, and in beta spindling associated with bipolar, very commonly they're given. But if the person has beta spindles and looks bipolar and they have discharges, you have to switch from these to a totally different approach to deal with the beta, and you have to use if there's a paroxysm that's overt, you're going to use a more potent anticonvulsant. And the tetracyclics can reduce beta, but they're given at night. They reduce the beta, they help you sleep, and they have an anti-anxiety effect during the day. Valproic acid is Depakote, and It's used for paroxysms. It's an older style of an anticonvulsant, and it does have liver issues. So blood testing has to be done consistently to make sure you're not causing a liver problem with it. Lamotrigine is really pretty well tolerated, but the one problem it does have is Stevens-Johnson syndrome. So if you start to take it and you get a fever or a rash, you have to stop it because it's your immune system acting up. And if you don't stop it, you can end up with aplastic anemia, which can be fatal. So if you get a rash or a fever, you just stop this. You can switch to a different kind of an anticonvulsant.
So can I make a
Ethosuximide.
comment on lamotrigine because I really like this
Yep.
as an anticonvulsant. So the rash or
Yep.
the fever, there's really some subtlety there. If the rash is associated with flu-like symptoms, the client or the patient is to stop the medication immediately. But if it's just a rash, basically you observe it and you let it go because lamotrigine is a very mild anticonvulsant in terms of side effects when I compare that with the other ones.
Yep.
So it is a fairly good medication to use for this purpose.
Yeah. If it's a 3 per second spike in wave, ethosuximide is a really good match to it. And 3 per second spike in wave is the absence style epilepsy, where it's a staring spell. When school starts, we usually get a referral of a whole bunch of kids that they suspect of having, staring spells, staring and blinking, and that's a perfect match. Keppra is ideal for temporal lobe discharges, and it's the most common anticonvulsant. If you go to an emergency room to be given, they're going to send you out after your ER visit to see the neurologist, but they'll send you out on Keppra. They would like to send you out on Lamictal, but the possibility of a Stevens-Johnson syndrome popping up between the time they refer you and the time you're seen. So Keppra doesn't have that kind of a side effect, and it's more common to be given in an ER setting. Clobazam is a benzodiazepine, is sometimes referred to as Onfi as a brand name, but the clobazam is used as a primary anticonvulsant in fairly severe cases. It's not generally handed out because it is a benzodiazepine and it has a street value in it, so it's very, very controlled. And then benzodiazepines in general can be given to stop an ongoing seizure, status epilepticus, and Diastat is a nasal or a rectal suppository, and the nasal one is easier to give in public, so I usually recommend that the parents acquire a nasal Diastat. So if their child's having a seizure in a public place, they can end up stopping it without having to use a suppository. If you're bedridden and at home, either one of them is a perfectly appropriate way. SNRI reduces frontal alpha and coherence. Now that's the intention, but it also speeds up the background slightly. If it's an SSRI, you get the effect without the speeding up of the background. Reduces the alpha power and coherence up front. And the coherence is a connectivity measure. We saw some of the connectivity source analysis in the database that I kind of skimmed through real quickly. And then tetracyclics reduce the beta, but they're sedating, rather strongly sedating, and can't be usually given as a daytime. They're given at night, and they have a longer half-life, and they have an antidepressant and anxiolytic effect during the day. So again, hormones given for social bonding is oxytocin, and oxytocin it's less commonly seen, but I'm seeing it more often as people become more familiar with the reason for it being given and the effect. And again, it reduces mu centrally, C3, C4, alpha hotspots, Mu happens when the mirror neuron isn't connected to the outside world. So if you're not relating to the social world outside, the mu on the right side lights up. If you're not relating to the language world outside, language and math, the left side mu lights up. And obviously, medications can reduce the mu, and they modulate the alpha and beta. And the publication for that is listed in the PowerPoint. Now, the photobiomodulation is a emerging application in ADD ADHD. Karosh, did you want to do this, or you want me to just finish this off?
Go ahead, Jimmy.
Okay.
There's a question first. If Carmen's asking if someone's already on the right medication, but they're doing neurofeedback or photobiomodulation, and that affects their medication, what are some things you can look for when to tell a client to talk to the prescriber about like medication monitoring when they improve with trainings? And then what do you tell a provider when they're not familiar with EEG, neurofeedback, or photobiomodulation?
Well, to start with, if you're going to be doing work with somebody who's being treated with a medication, you should be notifying the physician or the team that's doing the treatment, because they need to know that you're working with their client. You're now working with the same client. As you reduce the physiologic need for the medication, it's going to look like the meds are perhaps a little bit high in the dosage, and that they're going to monitor the meds. And if the patient looks like they're on too much of the medication, they can drop it. You're going to change the biological demand for it. And they need to be sensitive to the fact that you're changing the level of demand. And over time, that's going to end up making it look like they're on too much of the medication, and they can end up dropping the meds and reducing them. Now, the anticonvulsants, they're going to monitor if it's an anticonvulsant being given for epilepsy, they're going to monitor that periodically anyway. And we've basically just let them know that we're working with their client that has epilepsy, and that we're happy to have them manage the meds just the way they would normally. They do periodic EEGs, and if they see an EEG without a spike in it, they're going to think, well, maybe it's time to rethink the meds and reduce the meds. They'll drop the dose, do another EEG. If it's still clear, you might have walked away without the meds being on board anymore. We have a good track record of working with intractable epileptics and working with them to the point where they're seizure-free and med-free, and some of them for like 7 years. We just wrote up one on a client. Her name was outed in the paper deliberately. She's graduated with honors from Baylor University, did a 4-year tennis scholarship as a tennis star. And she's finishing her grad school now and intends to go pro in tennis and be a promoter of neurofeedback because she's escaped an intractable epilepsy with hundreds of seizures a day and incapable of even doing simple self-care. So, but let's talk a little bit about the photobiomodulation. To start with, it's shining light on the cortex. And you think, well, you've got a skull in the way. I mean, how can you simply shine a light on the cortex? Well, light actually penetrates the skull as translucent, and some of the light intensity actually makes it to the cortex. And the stimulation of the cortex actually ends up changing EEG content with the stimulation. So in the device, the iSync Wave device, they have all 19 channels. You can select 1 to 19. And so you can focus on one spot or do all of them. And you can pick from 1 to 45 hertz for the frequency that you're stimulating. And basically, you can design where you're going to shine the light and what kind of frequency that light's going to be flashing at. and the brain does some following. So if you flash it at 10, you get a 10. If you flash it at 12 or 14, you get a 12 or 14. If you flash it at gamma, the entire brain ends up getting stimulated at a mitochondrial level. So what you flash ends up being important. And again, hypoperfusion, if you see something that's got hypoperfusion, you need to stimulate it. We use the neutral positive perfusion. One of the things that happens with slow edge of alpha is that it can show ischemia, post-traumatic ischemia, as an example. And one of the things you don't want to do with ischemia is overactivate the area that's got the ischemia. There's an impaired function there, not enough oxygen and glucose are getting to the cells. That's ischemia. So if you do a lot of activity and make a large demand for function, you can actually create cell death. So we do a gentle positive perfusion, not creating hyperperfusion, just an above the mean. The brain regulates its own blood supply. If you turn on the brain area, it shunts more blood to that area. So it's going to open up the blood vessels in the area if you use the gentle activation. If you use overactivation, you can actually cause problems. So we look at this and design the intervention. Some people just need alpha. They have a low voltage fast EEG and they need more alpha. So you can stimulate at the alpha frequency and increase the perfusion and actually create some alpha. So what we've got is a ability to pick a frequency based on the perfusion. 3 ways that photobiomodulation has been shown to work. At a mitochondrial level, gamma frequency stimulation gives the mitochondria a boost, and that's beneficial for networks. Brain networks require cells to be hooked together, and the mitochondrial power house of the cell ends up being needed to end up having that cell available for the network. So having gamma stimulation, which is generalized, ends up helping any network that ends up having to form, because each cell has to be used in a variety of different networks. If you had to have one network for everything you did, we wouldn't have enough brain structure to have all that many networks. So networks use cells with different combinations. So the 40 hertz stimulation gives every cell a boost, and that allows them to be networked. So that's an important one. Um, Nitric oxide is essentially going to cause vasodilation. And when you've got ischemic problems, you want the blood vessels to open up. And rather than taking a medication that gives you nitric oxide, you can actually create the effect with the gentle positive perfusion stimulation in the SMR frequency range, giving you a gentle activation. And the glia, calcium channel signaling is a glial phenomenon, and calcium channels are in neurons as well. But the calcium signaling is influenced by photobiomodulation as well. So we've got the ability to help the brain by pointing at frequencies it needs to make in order to function better. The ability to counterbalance when something is not quite tuned properly. And again, the calcium signaling is a glial phenomenon. So the photobiomodulation ends up stimulating more than just the skin surface that it's hitting. It goes through the skull and skin and meninges and actually works in the brain. Now, you might say, well, show me the data. Well, here's a photobiomodulation study, and they've got a control group. And with the photobiomodulation, this is relative power in the delta band. So this is very slow activity in the brain, and it's not necessarily a good thing to be having. And in ADD that gets sham treatment, the delta actually went up slightly. Here in the actual active treatment with photobiomodulation, the delta went down, and the difference is significant. Now, this is basically looking at the reaction time. And reaction time went up slightly in the control group, the sham treatment. And it's an easy one to do a sham with. You can't tell whether it's actual or sham. You can't feel it, basically. And here in the treatment group, you can see that the reaction time is quicker. And that's a significant difference between groups. So the clinical impact of less slow content is massive in function, and having a quicker brain ends up being a reflection of proper function. This would probably be something you could see with event-related potentials as well, but the study here looked at delta power, relative delta power, and reaction time. The papers, every medication ends up having a publication associated with all the recommendations. So when we said something was related to dopamine or norepinephrine or so forth, there's papers to back all of that up.
All right, wonderful. We do have another question from Dr. Thompson, Linda Thompson, asking if you could comment on wavelength as a factor affecting efficacy for PBM.
Well, there are people that talk about the wavelength penetrating differently, but I think that has to be something that's got more studies than I've seen. I'm not a specialist in photobiomodulation. I'm a generalist in that area, and my specialty is EEG waveforms and the brain function, but the photobiomodulation has known effects. The difference between one device and another, they'll make claims that the frequencies that they're using are more effective than others. But again, I'd like to see more studies on that, and fewer claims and less claims and more studies.
Okay.
I would, Jay, I would actually confirm that in our clinical work, we work with different wavelengths with PBM, and from what we've seen, really there isn't a discernible difference in terms of efficacy. So I would say that yeah, there's definitely room for more studies on this, Linda.
And I do appreciate the studies being done with an independent group. Too often it's the vendor themselves doing it, and I appreciate the iSync publishing their studies in peer-reviewed journals and not just presenting them at a meeting and using them for marketing claims. So congratulations on keeping true to the neuroscience. But it's what I would expect. My friends, Dr. Kim and Kang, I've known for quite a long time. They've put up with me for over a decade, and that's not easy. So I appreciate them personally because of their dedication to the neuroscience. And I see far too many people that are in it for something quick and easy. They've taken their time. It's one thing to have a lid that you can set on top of your head. It's another one to have one that when you stretch it out, the 1020 system stays in the right places. So they've maintained true attention to the detail of the neuroscience and the engineering. And as you can see inside, this is on the area of where the forehead would be. I don't know if you can see my mouse. The three on the bottom, you can see how smooth they are. but the rest of them have to go through hair. Well, on me, quite a few of them don't have to go through hair, but nevertheless, the comb-like ones here end up having a polymer coating and silver chloride. So they're sensitive all the way down to DC or 0 cycles a second. So they're very accurate. And in the center of each of these is the photobiomodulation little LED laser to stimulate. So these are coated. They're although they're kind of bristly, they're not pointy and sharp, but they do go through hair. And for those of you who have it, having hair gets in the way of a lot of electrode applications. Dry sensors that are, you see some that have a solid metal. The ones on the forehead can be solid metal coating. They work okay, but the ones that have hair between them and the skin have contact issues. And if you don't have a well-engineered device, you just don't end up with a good contact. And in dry sensor technology historically has had difficulties with contact jitter, the contact being made and formed and disconnected, and that contact jitter has made most of them unable to be used clinically, and this device has gone through FDA. So it does a nice job. It's a clinical FDA medical device, not just not just some general health and wellness thing.
Thank you for presentation, Jay and Koroshu, and thank you so much for supervising our group. Thank you, Jay, about the mentoring so long time for developing this technology. And I'd like to briefly share a case study for the PBM ADHD child. This is the baseline EEG, as you can see here. This is the baseline EEG, and this is the clean data. And I want to show you the baseline. His EEG shows a lot of theta activity, 4 Hz excess in the frontal, and 6 Hz, and 8 Hz excess at the baseline. And in the spectra, we can see he has, for example, here, he has a 4 Hz rhythm, and also there is a 6 Hz rhythm and 8 Hz rhythm in the relative spectra at the baseline. We applied the photobiomodulation with the 13 Hz stimulation globally and also 15 Hz stimulation and frontally. And this is the baseline, and after the 4 weeks intervention, and we can see the improvement here. This is another recording after 4 weeks PBM intervention. And this is the data and the clean data. And I'd like to briefly show you the relative power map, and the 4 Hz slow wave at the baseline is disappeared, and the 6 Hz still appeared here. And we can see in the spectra, we can see here, this is the relative spectra. There is a 6 Hz still remained, but we cannot see the 4 Hz at the baseline. As you can see here, at the baseline, we have the 4 Hz and 6 Hz and 8 Hz, but during the intervention, we used the 13 Hz photobiomodulation globally, and also I combined the 40 Hz 10 minutes 13 Hz PBM, and after that the 5 minute 40 Hz stimulation, and then I have this kind of 4 Hz rhythm is disappeared, but 6 Hz still remained, and that's why we have this kind of spectra. And then another 4 weeks intervention here. This is another EEG recording. Sometimes the EEG channel, there is the quality is not good, but we can make the clean data using the denoising algorithm. And then after 8 weeks intervention, we can see the 4 Hz and 6 Hz. We cannot see that 4 Hz, 6 Hz rhythm and still having the 8 Hz alpha rhythm. And in the spectra, we can see this is also relative power. The 4 Hz and 6 Hz are substantially decreased, and there is only the alpha peak activity at the 8.8 Hz. At the starting, we have the alpha peak frequency is similar at the starting point, but we give these children this 13 Hz global and 40 Hz, and also we can give him the 15 Hz like this, the frontal activation using the photobiomodulation. At the baseline, actually, he cannot go to the school because of his violent behavior with his friends at the school setting. So school expelled these children. So he started to get our QEEG-guided photobiomodulation intervention. After 8 weeks intervention, he has a quite good EEG profile compared to the baseline, and then he returned to the school because school accept he can come to school because his EEG is calmed down and his behavior also can be controlled after the 8 weeks intervention. I think this is the case example, and we can tune his frequency step by step, and like the 4 Hz decrease, and after that 6 Hz decrease. Now he still has the 9 Hz frontal problem, but basically a slow wave is decreased when we use the 13 Hz SMR global stimulation and the 15 Hz frontal stimulation combined with 40 Hz stimulation. I think that kind of a step-by-step tuning of the slow frequency to the high frequency frequency band using the photobiomodulation can give him more control his frontal activity. I think this is kind of a case example I can share at the moment. So basically, the basic photobiomodulation mechanism is the mitochondrial activation, and also we can take advantage of this kind of frequency-tuned entrainment using the specific frequency the client should have, but usually they cannot make that kind of rhythm, but the right-gated ion channel in the neuron and the right-gated ion channel may be entrained by the frequency of the frequency-selected photobiomodulation. It is kind of pacemaker in that reason, because many assembly of neuron in the stimulation area can kind of entrainment using the right-gated ion channel in that area. They can firing together and making this kind of rhythm and tune step by step to make an improvement. This is the simple case study, one case study for ADHD. So you can take advantage of this kind of neuromodulation without any side effect, and QEEG guided, make a safe intervention for the children. Not just children, this is the specific ADHD intervention. We have a lot of different intervention study, so you can take advantage of this new digital neuromodulation technology. This is my case presentation. Thank you.
Thank you. It's impressive to see a clinical turnaround in such a short period of stimulation sessions, and such a nice normalization of the spectra. You're showing it in Z-score, so the 4 and the 6 have gone from red outliers to green norms. So, uh, and if you've gotten a kid to be able to be in school instead of being rejected from school, it can change the life. So I'm sure the family is quite happy.
Absolutely
Are you
amazing.
muted?
Yeah, thanks for sharing that, Piers.